I would just like to welcome everyone. You would have gotten this CNS Working Group agenda. We have a very robust program. I would like to thank all the speakers for their interest in our group. This is relatively a young working group, but we have had a lot of interest. And I would like to thank everyone for their support. And I think you'll find that we'll get much more momentum now between this meeting and the spring meeting. The hope is that by spring we'll be able to meet in person. So thank you all. We had all wished we would have been in person, but we're still able to meet virtually. So the CNS Working Group has been a recent addition to SWOG, and we've been working under the auspices of the Rare Tumor Committee. Dr. Razel Kazrock has been very kind to us to share the resources with us to help our working group until we can get a couple of trials approved. And then Dr. Blanky has said that our group would be considered for a full committee and with full resources. So thank you so much for all your input, suggestions. In the interest of time, I've got 10 minutes to give some updates. I'm going to talk about this Maverick study, which is being led by Dr. Chad Rastovan, who's obviously with us, along with Dr. Paul Brown. This is a study which is being led out of SWOG S1827. And essentially, we all know that we do PCI, or prophylactic cranial irradiation, in patients with small cell lung cancer, both in limited stage and extended stage, to decrease the brain metastases. And it has led to overall survival, as has been shown by 2 landmark studies. However, this is then has a negative aspect of neurological toxicities. The 2 studies that led to this practice changing effort were limited by heterogeneous or absent brain staging and surveillance. And as you can see, the publications were in 1999 and 2007. So these studies were done a while back. We've had significant changes in MRIs and how thin cut MRIs we have. So the question is, can we revisit it? There was a Japanese RCT, an extended stage small cell, that was published in Lancet Oncology 2017, evaluated surveillance plus minus PCI, reported no differences in progression-free survival or overall survival with addition of PCI. Median OS very comparable in the 2 groups, 13.7 months to 11.6 months. And essentially it was making us revisit this issue. So Doctors Paul Brown and Chad Rastovan are looking at this, and as you can see, these are the NCCN guidelines for small cell lung cancer, where prophylactic PCI is recommended for limited stage. For extended stage now, MRI, brain surveillance, plus minus PCI is recommended. So the question is, the jury's out there. And so with the latest compliance that we have with patients, the question is, can we do just MRI screening and not do the PCI? So this is the question through Maverick study that is being asked. Small cell lung cancer patients, both limited or extensive stage, those who have no prior brain metastases, no brain metastases on MRI after first-line therapy, will be eligible and will be randomized to PCI or no PCI. Stratification will be done with limited versus extensive stage, immunotherapy, yes, no, performance status. And the 2 groups will obviously have brain surveillance. Primary endpoint is overall survival. Secondary endpoints are cognitive function, OS in limited and extensive stage, brain metastases free survival. And there are a number of translational endpoints which are in the slide. We need 600 analyzable patients, which is the cruel goal. Hippocampal avoidance PCI and whole brain radiation are both allowed. Next, this is a study that is actually an effort being led out of NRG, which is NRG BN007. It's a randomized phase 2/3 open label study of ipilimumab and nivolumab versus temozolomide in patients with newly diagnosed MGMT, unmethylated patient population, which we know gets very limited benefit from temozolomide. This is asking the question of in this unmethylated patient population of the standard treatment. They are allowing Optune along with adjuvant temozolomide, and the experimental arm has ipilimumab and nivolumab instead of temozolomide. As you know, temozolomide only adds 24 days worth of benefit in this patient population. So most of the recent unmethylated MGMT trials have skipped temozolomide in the experimental arm. So this trial is enrolling through NRG, and SWOG has decided to also support and champion the study. So for members of SWOG who do not have this trial open, will have this capability. The phase 2 primary objective is looking at progression-free survival. Phase 3 is going to be looking at overall survival. Target accrual is 485 patients. The key eligibility criteria is obviously proven diagnosis of GBM by central path, MGMT, unmethylated status, and they are excluding the IDH mutation patients, so only will include IDH wild type. Dr. Craig Herbinsky is with us, and he's going to give us a presentation soon about the new WHO classification updates and how it will impact our clinical trials going forward. So then there's another trial. This is the Alliance study, which we have decided to champion. It's a study being led by Dr. Gavin Dunn from WashU. It's a phase 2 study of checkpoint blockade immunotherapy in patients with somatically hypermutated recurrent glioblastoma. Our CNS group had tried to see that if we could get a component of glioblastoma in the DART study, which is a study of ipilimumab and nivolumab in rare tumors. And we tried, but because this study was being approved by CTEP, our approval was, we were asked to actually become part of the study. So we have decided to champion this trial as well. And basically, this is the trial schema. It's for patients with glioblastoma here. Both wild type and mutant patients will be allowed. Patients in first and second recurrence will undergo surgery to confirm disease recurrence. And then the tissue could be sent to Foundation 1 testing to identify tumor mutational burden. Patients with TMB of more than 20 will then get a combination of Nivo and Ipi. It's a 2-stage design study. In stage 1, there'll be 18 patients. If 1 or greater patients get a response, stage 2 will be another 14 patients. And essentially, we know TMB has been associated with benefit in other tumors. There is some suggestion that there may be limited benefit in glioblastoma, but this study is going to ask this question in a prospective manner and will give us results whether dual checkpoint blockade is a good strategy in this patient population. As mentioned, the eligibility criteria is GBM in first or second recurrence. Essential component here is TMB of 20 on Foundation 1 testing. There has to be measurable disease because the study is based on response rate criteria. So the RANO criteria of at least 1 centimeter by 1 centimeter is used. Essential component is that patients should not be on steroids or dexamethasone of 4 milligrams or more. So anyone who's on low dose of steroids is going to be allowed on this study. We know steroids mitigate the impact of immune checkpoint blockade. And prior studies have shown that if patients are on steroids at least 4 milligrams or greater, do not get a lot of that outcome. So that's all I have in terms of updates. I know Dr. Rastovan has some updates on some of the brain metastases studies which are being done through other cooperative groups, and he'll have an opportunity to talk about some studies which are being led out of Alliance by Dr. Paul Brown and out of NRG Oncology by Dr. Vinay Gandhi and colleagues, which are being championed through SWOG. But definitely, if you have questions for a Maverick trial that is being led by Drs. Chad Rastovan and Paul Brown, you will have opportunities in the final session when he'll speak about some of the brain met study. So that's the input. We also are going to start having regular meetings of the CNS Working Group. So stay tuned. What I would like is in the chat box, please put your emails so that we can add you to the email chain as we set up these meetings. And we look forward to robust interaction. With that, I'll move on with the agenda. And we have Dr. Craig Herbinski, who's internationally renowned neuropathologist from Northwestern University. He's going to talk to us about WHO 2021 classification of CNS tumors, implication for clinical trials. Thank you so much, Craig, for speaking to us.
Okay, thank you very much for inviting me. All right, the neuro-oncology textbook, the new textbook, the 2021 scheme was supposed to have, it was completed quite a while ago actually now. It was supposed to have been released in print form this past July. For reasons I guess that have to do with the COVID supply chains, etc. I am not clear as to what's going on, but the official textbook is not yet available. However, this summary was issued over the summer in Neuro-Oncology. It's a really extensive and very good review article in it, and it certainly does codify everything and summarize everything that is in the new 2021 WHO book. So if you need to, you can certainly look at this and reference this as needed. There was another thing that came out in companion to that. It was a really nice little short letter written by Patrick Wen and Roger Packer on the clinical implications in particular of the new 2021 scheme. And so I'll be distilling both here in the next 20 or so minutes, in particular what is most relevant for the new clinical trials. This is a mock-up of the new cover of the CNS book, or whatever it's worth. This is actually the 5th edition of the CNS, and it came out, the last one was in 2016, and this one came out earlier than was originally planned by the WHO publishing group. But because of the rapid changes in molecular classification and diagnostics, it was felt that this book was going to jump ahead in the line, so to speak, and be issued sooner, only because there's so much out there now that needs to be done on a routine clinical basis. One change that I really, really liked, no more Roman numerals for WHO grading. For a lot of reasons, one of which was too easy to mistype as a pathologist. It was also too easy to misread in path reports, especially the difference between a 1 and a 2 or a 2 and a 3. So now it's all Arabic numerals. And really, I think the crux of this is the issue of lumping versus splitting. So we've got, in the evolution of Homer Simpson and the Simpsons there, at what point do you, here you got these different stages of proposed evolution. And at what point do you just say, they're all Homo sapiens, or they're all primates, or they're all this, or they're all that? How far do you parse things out for the sake of accuracy, but at the risk of losing a big cohort for clinical studies? Well, this is the new 2021 scheme. These are all the tumors. These are all the tumors in the 2021 book that are defined by specific genetic alterations. The alterations have to be there for the diagnosis to be rendered. We're not going to go through each of these line by line, but it just does underscore how important molecular testing is now. Not just testing for a handful of markers, but really a good panel that screens for a lot of things. It's not just about IDH mutations anymore. Now we got diffuse MIB1 gliomas, mostly in kids. We've got weird things like the CNS neuroblastoma that's defined by activating mutations in FOXR2, B-core internal tandem duplication of some very rare tumors that, unless you're at a high yield coordinary center, you're probably not going to see them too often. Here's all the newly recognized tumor types, most of which are indeed defined by molecular criteria, meaning the molecular alteration is part of the diagnostic line now. So for example, we've got diffuse hemispheric glioma, which is H3G34 mutant, spinal ependymoma that's Mcn amplified, so on and so forth. New grading scheme. So for IDH mutant astrocytomas, now and we'll get into this in just a moment, grades 2, 3, and 4. It's grade, so in other words, no longer are we calling anything a glioblastoma that has an IDH mutation. If it has an IDH mutation, it's either an astrocytoma or an oligodendroglioma. The term glioblastoma is strictly reserved for IDH wild-type diffusely infiltrative gliomas that have other molecular and histologic features. Now, one thing that was especially helpful, I think, in terms of conceptualizing these gliomas and the glioneuronal tumors was reorganizing them into adult-type diffuse gliomas, which are your IDH mutant astros and oligos, and then your IDH wild-type glioblastomas, and then splitting pediatric-type gliomas into your diffuse gliomas into low-grade versus high-grade tumors. And then we've got circumscribed astrocytic gliomas, which include mostly BRAF-driven tumors, PXAs, pilocytics, that sort of thing. And then we've got glioneuronal and neuronal tumors, and finally ependymomas. So again, this adult type diffuse gliomas, I think that it's a lot simpler now in many respects. Like I said, you've got, if it's an IDH mutant astrocytoma, it's going to have one or more of these other alterations in addition to an IDH mutation, and you grade it based on both a combination of histology as well as this molecular. So if you have, for example, a grade 2 or 3 tumor, but it has homozygous CDKN2AB deletion, that's tantamount to a grade 4 diagnosis. You can upgrade the tumor based on that alone. Oligodendroglioma, it now has to have an IDH mutation and one P19Q co-deletion. Right? And they almost always also have a TERT promoter mutation, the same kind actually, ironically, as occurs in IDH wild-type glioblastomas. And you can also have these other alterations. Notch1 in particular has been associated with worse outcome, although it doesn't yet officially equate to a grade 3, but there is no grade 4 oligodendroglioma just yet. Glioblastoma is now, it's this classic thing, of course, that's what we encounter most often, and that's still going to be grade 4. Necrosis and or microvascular proliferation in an IDH wild-type tumor. It has to be an IDH wild-type tumor. Again, if it's IDH mutant, you don't call it glioblastoma anymore. So there's no such thing as an IDH mutant glioblastoma. That would be an IDH mutant grade 4 astrocytoma. And typically they'll have these other things. So especially if you have a histologic 2 or a 3, and you pick up a TERT promoter mutation, gain of 7, loss of 10, and or EGFR amplification, that is considered sufficient to be calling these IDH wild-type glioblastomas. And actually quite often that ends up being the case for us. We upgrade a lot of tumors based on these molecular features. And this is just a Cancer Genome Atlas summary of taking all these tumors, all these diffuse adult-type gliomas, and stratifying them. In panel A, it's all the tumors in the Cancer Genome Atlas assorted by the presence of IDH mutant and co-deletion, IDH mutation by itself, meaning an astrocytoma, or IDH wild type, which are glioblastomas. You can see how cleanly they sort out. Panel B are all the IDH wild type glioblastomas that are sorted by histologic rate. In other words, how it looked under the microscope. And you can see there really isn't that much of a difference. In panel C, these are all the IDH mutant astrocytomas, meaning they don't have one P19Q co-deletion. And there is a difference in median survival between a grade 2 and a 3, which is characterized by increased mitoses and nuclear atypia, as well as histologic grade 4, which is the presence of necrosis and or microvascular proliferation. So histology still does matter. And in panel D are the oligodendrogliomas sorted by histologic grade 2 versus 3. So histology still does have a role to play in the classification scheme. What's interesting, though, just for whatever it's worth, is now everybody's locked in on the new 2021 WHO book on homozygous CDKN2AB deletion. For example, these are all in IDH mutant astrocytomas. Homozygous CDKN2AB deletion does equate to a grade 4. But even this loss of just one of the 2 alleles, in other words, hemizygous loss, it does associate with worse survival, worse overall survival than if the gene was completely intact. So I think over time we're going to see both maybe in astrocytomas and in even meningiomas, hemizygous loss of this tumor suppressor gene. Hemizygous loss may end up being associated with like a grade 3 diagnosis, maybe not quite grade 4. As far as pediatric type diffuse gliomas, there's been a lot of changes, more so than in the adult tumors. There's been a lot of changes relative to 2016, and that indicates just how much that particular field has advanced with molecular. We don't have to go through all of these, except just to note that they are characterized by low grade and high grade. And it's important to note that an adult can develop a pediatric type diffuse glioma. In other words, I've diagnosed plenty of older people in their 40s and 50s even with diffuse hemispheric gliomas that are histone G34 mutant. Same goes for the midline gliomas. I mean, I've seen them in as old as 70-something years. They're still considered pediatric type diffuse gliomas, even if the tumor arises in an adult. Conversely, if a child develops an IDH mutant tumor or an IDH wild type glioblastoma, if they're like in their mid to late teens, that still counts as an adult type diffuse glioma, even though technically the patient is a child. Circumscribed astrocytic gliomas didn't undergo a whole lot of changes, except they are now broken out in their own different subgroup, which I think is very useful because they're not diffusely infiltrative, so they have a different clinical course. One that is worth noting is this new addition, high-grade astrocytoma with pyloid features, which is basically like an anaplastic pilocytic astrocytoma, but it's got hits in ATRX and CDKN2A and B. We don't have to spend too much time on glioneuronal tumors, except to say that there are a couple of new entities that have popped up that are extraordinarily rare. Some of these are very, very rare tumors, but they're nonetheless distinct enough because they have distinct molecular alterations. Very often they have a distinct histologic appearance, and they have a unique clinical course. Although broadly speaking, most of these tend to be fairly indolent, but otherwise there really wasn't a whole lot of other changes. Ependymal tumors, this is another one that's undergone a fair amount of shifting. So now among the supratentorial ependymomas, they can either have ZFTA fusions, which used to be called RELA fusions. Now they're ZFTA fusions and YAP1 fusions. ZFTA are supposed to be a little more aggressive. YAP1 are less aggressive. Posterior fossa, the big split now, in both in kids and adults. If you see a posterior fossa ependymoma, you're supposed to, as best you can, subtype it according to whether it's group A versus group B. And even though histone trimethylation has been proposed as splitting the two apart, in my experience, whole genome methylation profiling is your best bet. That's going to be your most reliable way to split them. Spinal ependymoma, there's two flavors. You've got your NF2 one that are characterized by NF2 loss. And those tend to be extraordinarily indolent. They have very, very good 10-year survival. But the ones that have MYC-N alteration, usually MYC-N amplification, they're the ones that are much more aggressive, and they tend to disseminate throughout the cerebral spinal fluid. Myxopapillary ependymoma has been upgraded from a grade 1 to a grade 2 because there's too many of these tumors that do behave, at least they tend to recur a little bit more. Rarely they'll disseminate, but not too often. Subependymoma is still a grade 1. No changes there. Embryonal tumors. This was a tough section. This is one of those where the histology of these tumors can have so much extensive overlap, especially when they occur in the supratentorium of either kids or even adults. It's virtually impossible to reliably subtype these now according to the new scheme without having some form of molecular alterations to go with it. So again, CNS neuroblastoma that's defined by FOXR2 activation, or B-core internal tandem duplication, all these other alterations that are associated with certain high-grade embryonal tumors, they all tend to be very bad actors, bad outcomes. Medulloblastomas, they've got now, of course, we still have the Wnt activation, sonic hedgehog, which is really driven by either the presence or absence of P53 mutation. So if it's got a P53 mutation, that's a very bad prognosis. It's almost as bad really as like the MYC-N driven medulloblastomas. And then you got this non-Wnt, non-sonic hedgehog, which used to be called group 3 and 4 tumors in the original schemes. They've combined them because there's really not a lot of evidence to indicate that they're group 3 versus group 4 do any different, except that if you've got alterations, again, in this MYC or MYC-N gene or isochromosome 17q or something like that, then you're dealing with something that's going to be a lot less pleasant and much more likely to disseminate throughout the cerebral spinal fluid. Meningiomas. All right, so this is an emerging thing here with the classification of meningiomas. And we don't have to go through it in extraordinary detail, except that a couple of mutations do seem to segregate with particular behavior. SMARCA1 alterations in the clear cell subtype, which is a grade 2. And then you got BAP1 and rhabdoid and papillary subtypes. That's grade 3. But the most important thing, at least for meningiomas routinely, is to try to screen them for the TERT promoter mutation and CDKN2AB deletion. And if you have either of those hits, then it's tantamount to a grade 3, regardless of what the tumor looks like. And there's a lot more that's emerging now in terms of other chromosomal hits like 1p loss or methylation clusters. Those all still are, and that's an evolving field. That's a little more fluid. Now, as far as implications of all this for clinical trials, well, you can see the advantages of this new scheme, and incorporating more and more molecular. It does give us a more accurate diagnosis, which relates, of course, to much more reliable prognostic stratification. So we're not overcalling tumors or undercalling them or misclassifying them. You've got a clear sense of what these tumors actually are. And in most cases, that's being driven by molecular alterations. And so as a result, then there's a lot more consistency, especially if people are using roughly the same kind of methylation profiling, the whole genome copy number analysis, and all their next-gen sequencing panels have the similar hotspots that are being screened. Then you get a lot more consistency from one group to another and between pathologists, because I mean, our inter-observer reliability based on just histologic impression, unfortunately, tends to be pretty low. And as a result, then, when you're enrolling patients and you're setting up clinical trials, you can develop much more homogeneous patient cohorts because the molecular criteria are there and either the tumor has it or it doesn't. But there are disadvantages. One is that there's a lot fewer trial options for these less common tumor types and types and subtypes. For example, grade 4 astrocytomas, these IDH mutant tumors now. It's going to be a lot harder to accrue for those relative to IDH wild-type glioblastomas. And the embryonal tumors is going to be a real mess too. I mean, it's going to be very, very difficult. A lot of these high-grade embryonal tumors that need better therapeutics, it's going to be hard to test them because these tumors are so rare. These specific subtypes are so rare. We also, as a result, are going to need a lot more comprehensive and faster screening on all these cases. And that feeds into this problem with third-party payers very often being resistant to this. In particular, the Center for Medicare Services has this 14-day delay where if you're an in-house patient, or if you're a patient and then the pathology specimen is sent to an external lab or other institution for more advanced molecular profiling, if it was a 14-day window between when the patient was in-house as an inpatient versus when the testing was done, then Medicare won't cover it. And a lot of third parties won't cover it either. Other payers won't cover it. And that's becoming a real problem because obviously we need to enroll these patients on clinical trials quickly. So we're working on trying to get that changed and have that 14-day delay either greatly shrunk or just disappear altogether. Because a lot of times you just can't make these diagnoses without this. And finally, it doesn't, the new scheme still does not take into account germline abnormalities. I mean, if somebody's got neurofibromatosis 1, maybe their tumor doesn't behave, their meningioma doesn't quite behave like a truly sporadic meningioma, or I should say NF2, but anyway, we still don't have that taken into account. But there are a couple of qualifiers, just FYI. And this is actually a very helpful one, it's not otherwise specified, because a lot of centers still are not going to have this kind of molecular workup available, especially globally. So this qualifier has been added to indicate, hey, we're calling it diffuse astrocytoma, not otherwise specified, because we really don't have the molecular markers that are needed to really classify it according to the current 2021 standard. So you'll see that coming, at least you should be seeing it coming from a lot of community settings and maybe even overseas in certain countries. This not otherwise specified qualifier at the end of the histopathologic diagnosis. And then there's not elsewhere classified, which the confusion with not otherwise specified is understandable, but all it means is we did the molecular workup on this thing and it just didn't help. So sometimes you get once in a while these mysterious tumors that look low grade maybe, and then you throw a lot of spaghetti against the wall and nothing sticks and you don't know what to do with it. So in those kinds of settings, we can use the term not elsewhere classified. All right, that's all I have. Looks like I finished right on time. If anybody has additional questions, I either address them now or by email or whatever. Yeah, whatever's your preference.
No, so thanks, Craig. Such an excellent overview. So I have one or two questions, and I would actually encourage people if they want to ask, because again, I think this will impact our field. So one question I have is based on what you showed. What is the sense of what's the percentage of tumors which are really being profiled to meet all these criteria?
You mean nationwide? Well,
Yeah,
that's a great question.
I would say if you want to tackle that question, obviously at Northwestern, how you're doing, but also
at Northwestern, I mean, we're doing the pretty rigorous screen on every single case, and in fact, starting in November, we're doing in-house methylation profiling on every single tumor prospectively. And that replaces our current copy number assay because we don't need that anymore. It also replaces our current MGMT promoter assay because methylation covers that as well. So the whole genome methylation profiling. So we're doing everything on everything, but we are a coronary care center. That's kind of expected for a place like this. And as a result, we are getting a lot more consult cases from elsewhere in the region and even out of state. But we're running into this 14-day window with CMS where billing and reimbursement becomes an issue. So the bottom line is, I think as time goes on, we're going to have to see this, and it's not practical for every center to have this kind of testing in-house, but it is becoming, the data just support it being required. I mean, if you're going to do a 21st century workup, that's what's needed. And I think you're going to see more of these tumors get concentrated in a relative handful of centers nationwide.
Yeah, and what's the sense overall, though? I mean, in terms of interacting with top neuropathologists nationwide, what's happening? Because again, we all feel that brain tumor patients should be treated at multidisciplinary centers of excellence, but there's still a lot of patients get treated in the community. So in terms of when you are seeing this, what percentage of patients are you seeing up front, or what are referral cases that are being sent to you? Can you give us a sense?
Yeah, well, it's hard for me to say exactly what percent of patients nationwide, of brain tumor patients nationwide, are getting the what would be considered the gold standard workup now. I don't know I can tell you that, but I can tell you that since I started at Northwestern in 2015, I'd say about 10% of our cases were external consult cases. Usually people coming in for clinical trials, and they needed their diagnosis confirmed or in some way modified. Now we're probably seeing around 60% or so of our cases in-house, or of our cases that we see in our division, are coming from the outside, both for clinical trial enrollment as well as just a community practice saying, I have no idea what this is, and what do I do with this? So
I know there's a question from Dr. Jacob Mendel.
Okay, that's a great question. I can see. So would an IDH grade 2 tumor that lacks features of GBM still be considered a GBM IDH wild type? Well, those are the kind of tumors you have to be really, really careful with, because it's going to go one of two directions. It's either going to end up probably being a grade 1 tumor if it has grade 1 molecular features, or it's going to be the grade 4. So you can't just the absence of the IDH mutation by itself is not enough to call it a GBM. You've got to have the lack of the IDH mutation, and you got to screen the whole thing. So you got to pick, immunohistochemically negative is not good enough. You've got to screen it for the non-canonical mutations as well. So then you'd have to have a presence of TERT promoter mutation, gain of chromosome 7, loss of chromosome 10, and or EGFR amplification. And if you hit any of those criteria, then you're justified in calling it a GBM, full stop, IDH wild type. On the other hand, if you do all that molecular testing and you come up with an isolated like BRAF V600E with no other hits or anything, well, then you got to take a step back and think about it, because it's probably a grade 1 tumor. And that's where that, again, and just like what Dr. Odi was saying, that's where having people who do nothing but this for their living, it really helps, because we see those sorts of tumors a lot more, and we have a certain experience that causes us to maybe be a little more cautious. You don't just overcall tumors needlessly.
No, thank you so much, Craig. That was very helpful. I'm sure people may have questions for Dr. Herbinski. They're more than welcome to reach out to him. He's always such a great speaker and keeps people invigorated. So thank you, Craig, and look forward to interacting with you. With that, we'll move on. The next on the agenda is Dr. Tony Wang, who's a professor at Columbia University, and he's going to talk to us about his concept, an open-label phase 2 trial of Tocilizumab and Tocilizumab with radiotherapy in patients with newly diagnosed MGMT unmethylated glioblastoma. Tony, you're on mute.
I'm sorry, I was having trouble clicking the button. Can you see my screen?
Yes, we can. Thank you.
Okay. Well, first off, thanks again for my colleagues at the SWOG CNS Working Group for giving me the opportunity to present my concept. This is a phase 2 study of Atezolizumab and Tocilizumab with radiotherapy in patients with newly diagnosed MGMT unmethylated glioblastoma. And these are my disclosures, and they're electronically available. So, as most of us here in this working group know, glioblastoma is the most common primary brain tumor, accounts for over 50% of primary brain malignant tumors, and it's also the most aggressive. The median survival with standard therapy with radiation temozolomide is about 14.6 months versus 12.5 months with radiotherapy alone. And we know that most of the benefits of adding temozolomide is driven mainly in the subgroup patients who have the methylated MGMT promoter. So the MGMT methylation is prognostic for median overall survival in either arms. For patients that have the methylated MGMT, which is about a third of our GBM cohort, median survival is about 18 months. And for the unmethylated MGMT, which is approximately 2/3 of the GBM cohorts, it's about 12 months. It also serves as a predictor of benefit from temozolomide. So with methylated MGMT, temozolomide has been shown to increase the median overall survival by about 8 months. But in the unmethylated MGMT, it only increased median overall survival by less than 1 month. And I want to bring that up to your attention because with more of the recent studies out there with respect to GBM molecular analysis, we know that, for example, in a study from Northwestern by Lamano et al., in the TCGA cohort, that when you do RNA sequencing as well as microarray data analysis, IL-6 expression is associated with the worst overall survival in gliomas. Both of these are IL-6 expressions in the TCGA cohort, showing that higher levels of IL-6 expressions tend to be associated with a lower median overall survival versus those with low IL-6 expression levels. So this is a study from a group, I believe in Penn, that looked at specifically IL-6 induction or anti-IL-6 use that helps reduce the burden of M2 macrophage expression levels. And it's also mediated through a proliferator activated transcription activation of hypoxia-induced factor 2 alpha. So this is just an example of these anti-IL-6 mechanisms in terms of reducing burden of M2 macrophages. And then using these, this is another study from the same group that using genetic mirroring GBM models, this group here at Penn showed that specific inhibition of IL-6 and also specific knockouts of IL-6 inhibits macrophage alternative activation and also improves survival in these GBM-bearing mice. Here you have a IL-6 antibody and survival in orthotopic GBM mirroring models. The control group is the blue line in the survival curve, and then those that got IL-6 as well as combination therapy of IL-6 CSF1 showed improved survival, but certainly far better in the combination, suggesting that the targeting endothelial IL-6 may offer a selective and efficient therapeutic strategy for glioblastomas. So this is a paper from Lamano and Northwestern looking at anti-IL-6 therapies, and it's been shown that IL-6 signaling in GBM reduces both the local as well as systemic myeloid-driven immunosuppression, and enhances the immune-mediated anti-tumor responses against glioblastomas. This is actually one of their animal study model experiments where they had these C57BL6 mice who had first the intracranial implantation of the CL261 glioma cell lines, and then began antibody treatments with anti-IL-6 as well as anti-PD-1 just to test for its either synergistic or non-synergistic effects in glioblastoma. On the bottom left in that figure labeled J, you can see that the control group and the isotype, really there was no difference in terms of the change in terms of the tumor shrinkage, but then in either the monotherapy as well as combination therapy of IL-6 and PD-1, there's actually a reduction in tumor volume. And that's shown in the middle column in the bottom row under J, where the tumor volume changes were significantly reduced with the combination IL-6 and PD-1. And on the bottom right is the survival curve, which showed that the combination of IL-6 and PD-1 had improved survival in these animal models versus monotherapy, and certainly more so than the control group. So I'd like to highlight this study from a group in China that looked at Tocilizumab, which is a IL-6 inhibitor, and its ability to promote apoptosis in these U251 GBM xenograft models. And in this study, I think they did, I don't know exactly the number, but it was approximately 20 mice and they showed that the blockade of IL-6 receptors by Tocilizumab, demonstrating a certain level of therapeutic efficacy in the xenograft model. You can see that both in the tumor volume in the bottom row, where adding Toci IL-6 inhibitor was associated with reduction in tumor volume, and then when they looked at the IL-6 in the serum, it was also significantly reduced versus the control group. So going back to the studies by our colleagues at Northwestern that did the combination PD-1 IL-6 inhibition, I kind of expanded this survival curve here to show that the combination of PD-1 and IL-6 inhibition was associated with additive anti-tumor effects in these intracranial GL261 tumor-bearing mice. The survivals in the combination was approximately 55 days versus about 25 to 30 days in either monotherapy alone, and then certainly far better than the control group. So, I presented this at the spring SWOG meeting, and it was very helpful to present that because I got a lot of great feedback from a lot of our colleagues here and some of the members from the audience. So this is the proposal, which is a phase 2 trial looking for newly diagnosed glioblastomas to get combination atezolizumab and tocilizumab and radiotherapy. Patients will get maximal safe surgery up front, and only in the patients who have the unmethylated MGMT cohort, and that would be 2/3 of our GBM cohorts, will get radiation therapy in combination with atezolizumab and tocilizumab. The doses here that I have right below, so that schema is consistent with some of the current ongoing trials, including NRGBN010, I believe, followed by adjuvant atezo and tocilizumab. And the primary endpoint here is 1-year overall survival, and we project a improvement in 1-year overall survival by 70%, which will require a patient about 37 for 80% power and alpha 0.05. And we expect that this would take approximately 18 months to accrue and 24 months of follow-up. And then I'd also like to add that in terms of prior safety data, there are currently two studies that have already published on the safety of temozolomide and radiotherapy. One is at MD Anderson, and currently another atezo-toci combination for prostate cancer, as well as the use of non-small cell lung cancer. Again, the benefit of having this presented at the spring meeting was additional exclusion criteria that was also recommended by our colleagues at Genentech and also colleagues who have prescribed these kind of immunotherapy drugs, was to exclude patients with active infections, which is what they have used for their rheumatoid arthritis trials, for any sort of known active history of recurrent infections, And then similarly to currently the activated NRG-BN010 trial, which is using atezo, toci, and fractionated stereotactic radiotherapy, is also to excluding patients who have a history of autoimmune diseases, not limited to systemic lupus or rheumatoid arthritis and other autoimmune diseases. So secondary endpoints is to evaluate the safety of adding the atezo and tocilizumab to radiotherapy. As I mentioned, there's only one study from Anderson that published atezo, temozolomide, RT, and there's currently 2 ongoing trials for prostate and non-small cell lung cancer for that combination. We'll also look at progression-free survival, and we will allow bevacizumab option, and that certainly may need to be looked at separately for the PFS for the Bev and non-Bev patients. And of course, other exploratory endpoints are quality of life and tracking steroid use, especially in immune disease with these immune drugs. And then future options would include looking at peripheral blood immunologic profiles as well as overall survival toxicity. And certainly, of course, we'd love to explore biomarkers looking at the PD-L1 expression, IL-6 expressions in these pre-treated tissues, which may predict for efficacy to these combination arms, both in the OS and PFS variables. So in terms of accrual, this study essentially mirrors some of the multiple ongoing as well as completed studies. So both for the Checkmate 498 as well as the ongoing NRG-BN007 for newly diagnosed glioblastomas that omit temozolomide, for GBM patients without MGMT promoter methylation. Omitting temozolomide in both of these was not and has not been a barrier to accrual. Steroids are only given if clinically indicated, and these follows precedence of prior dual inhibition. So, so this, again, following the prior unmethylated MGMT cohort, no temozolomide, no placebo, and certainly without the temozolomide, it avoids the T-cell depleting effects of TMZ in the experimental arm. And because this is an unmethylated MGMT cohort, it certainly, it turns out will shorten the follow-up versus our methylated counterpart. So with that, I'd like to thank you for your time. Especially want to thank all my colleagues at the SWOG CNS Working Group, and especially my colleague and mentor, Dr. Fabio Iwamoto, for his advice on this concept as well. So thank you for that.
Thank you so much, Tony, for your concept. Since our last spring meeting, we actually I was able to present this at the Rare Tumor Committee, where actually it got good support. One of the things that was suggested to me was that the preference is not to do phase 1 studies, but rather phase 2. And then I did have some initial interactions with the statisticians on the trial design. So I think definitely there is support for this. And I think the next step basically will be a meeting with the statistician and also interactions with CTEP and try to see if we can get a pre-submission call with them just to see, because I just presented a whole volume of data, not only from some of the work that you, Fabio, and your colleagues have done, but also work from Northwestern, obviously work from UPenn, and an ongoing trial in the NRG's oncology space. So I think it's clearly of major interest to us as a working group, and also support from Dr. Kurzrock and the Rare Tumor Committee. So I think clearly let's get on a call maybe in the next week or two with some critical stakeholders and develop this concept, put it on the LOI of the SWOG, and then move this forward. But so thank you so much. I think the critical question will be, should we just do a phase 2 single-arm study of 37 or so patients as you have outlined, or is the data exciting enough to even potentially support a randomized, phase 2 trial? Hopefully, trying to still keep it under 100 patients so we could get it through CTIP. But I think that, for me, are the logical next steps. So thank you. At this time point, I would also want to open the floor for any questions for Dr. Wang. If not, thank you, Tony, and a lot of excitement about your concept. So thank you to bringing it through SWOG. It might be the first trial that might go through the CNS Working Group of SWOG. and we got to keep pushing our momentum. So thank you. If you want to share your screen so I can start sharing mine.
Yeah. All right, let me stop sharing.
Thank you. So I'm going to follow Dr. Tony Wang because, again, I think as you heard from Dr. Herbinsky, we have seen changes in GBM. We do realize it's not a single patient disease, it's very heterogeneous. And we clearly have definitely some biomarkers in gliomas which are able to be done and are important because that impacts outcome. So this is a concept that I've been working on with some colleagues here. It's a concept of TRC102 and temozolomide in patients with newly diagnosed MGMT methylated glioblastoma. And I think the great aspect of this concept, along with Dr. Tony Wang's concept, is they'll be supportive. So if our patients get new diagnosis of a glioblastoma, and going forward it's going to be IDH wild type, they will either be methylated or unmethylated. There's a very small portion which is indeterminate, but we would like to have trials for all of our patients. So all of you are aware of glioblastoma, and so I won't belabor the point, but it's the most common kind of tumor that we see, malignant tumor that we see in the United States. As Dr. Wang outlined, outcomes are still poor. On an average, our patients live, between 15 to 18 months. Survival is better in MGMT methylated tumors compared to unmethylated. Survival overall is still poor. Less than 5% of our patients survive 5 years. And I think the critical point is, as you know from NCCN guidelines, the preferred treatment for patients with glioblastoma is actually consideration of a clinical trial, which is actually rated even higher than standard of care treatment. So hence, there's a lot of aspect, and this really slide exemplifies that. I think we are one of the rare tumors where clinical trial consideration is placed higher than standard of care. Most of the other cancers, and we still have a lot of work to be done in cancers and getting the best treatments to our patients, but this is certainly more true of glioblastoma than any other tumor because we only have one chemotherapy that has ever shown a survival benefit, and then we have this modality called alternating electric field therapy, which has shown survival benefit, but has a very limited uptake in the patient population overall. So we have a lot of work to be done, and we all know that our patients, when they get diagnosed with this disease, are looking for clinical trials. So this is work from Dr. Rajesh Stoop, and again, all of you have seen this before. So this was the study that actually got temozolomide FDA approved, and this basically lent itself for support for combination of chemotherapy as well as radiation in management of these patients, followed by 6 cycles of adjuvant temozolomide. And the benefit was 2.5 months overall. I think one of the most exciting things for people who were treating these patients, at least at that time point, which was almost 2 decades back now, was the 5-year survival of the combinatorial approach, which was 10% compared to 2% in the radiation alone. However, as Dr. Wang had pointed out, there's a big dichotomy of the benefit, right? So we have these MGMT methylation marker, which basically is a repair enzyme. And if you are unmethylated, that means you actually have the repair enzyme turned on. There's a very limited benefit here. As you can see, the green curve is better than the yellow curve, but barely. So this is a 24-day benefit. So it's less than a month. So it's not really clinically that meaningful. And as Dr. Wang pointed out, number of recent trials, including Checkmate 498 and ongoing NRG study BN007, and several other trials have omitted temozolomide in this setting, and I think that has been well accepted. On the flip side, MGMT methylated patients got durable benefit with the combination. Here you can see the green curves doing better than the yellow curves, and definitely there is a 6 to 8 month benefit in this patient population, and hence we give our patients temozolomide and radiation. So meanwhile, we had actually run a study out of the Adult Brain Tumor Consortium, and several people were involved in this effort, and you can see their names and institutions on the slide. And essentially, this was looking at a study of TRC102 and temozolomide. And essentially, the premise for this study was that we know when we give temozolomide to any patients, it leads to 0,6-methylguanine, N-methylguanine, 7 and 3-methyladenine DNA adducts that are formed. We do know that the 06-methylguanine adduct is repaired by MGMT. But there's another pathway called the base excision pathway, which is important because the other two adducts of 7-methylguanine and 3-methyladenine are repaired by this pathway. We also know that N-methylpurine DNA glycosylase, or MPG, is the first step in the base excision repair. And this was a drug called TRC102, or methoxamine, was a drug actually invented by Dr. Stan Gerson, who was the Cancer Center Director of Case Comprehensive Cancer Center. And it basically works by binding to the apurine site created through the action of N-methylpurine DNA glycosylase. And this was work done by Dr. Andy Sloan at Case Comprehensive Cancer Center, showing that a combinatorial approach of TRC102 and TMZ, or temozolomide, led to best outcomes in orthotropic xenograft models, where he looked at glioma stem cells. A lot of people believe that glioma stem cells are responsible for chemotherapy and radiation resistance, and we have Dr. Justin Lathia amongst us today. He's going to talk about some of the work he and his lab are doing, and he's been a trailblazer in several of these efforts in glioma stem cells. So this led to this study that we ran out of ABTC, as I said, with a combinatorial approach of TMZ and TRC102. This was for patients in first recurrence of glioblastoma, those who were Bev naive. And this was a study which was done under the auspices of CTEP. However, we were put a big bar. The study was set to test the hypothesis that the combinatorial approach would achieve a 30% response rate. Those of us who have treated glioblastoma know that our response rates in recurrence is only around 5% at best with lomustine, which is one of the most commonly used agents we have. So this was a two-stage design study. First stage was 19 patients. Second stage would have been maximum 33 patients. And as you can see, the clinical characteristics were no different than what we have seen in patients treated on trials. Overall median survival of this approach was 11 months. Unfortunately, no responses were seen. Overall median progression-free survival was 2 months, again, comparable to what we've seen otherwise. Progression-free survival rate at 6 months was 10.5 months, again, very much within the realm of trials that we've seen in this modality. However, two of these patients that actually I had the privilege of treating on this trial did really well. One of them had a progression-free survival of 18 months plus, and the other one had a progression-free survival of 30 plus months. In fact, went out to almost 33 to 35 months. And both of them had a long OS. We then looked at correlative studies. This was work led by Dr. Lisa Rogers and the group at University Hospitals and Case Comprehensive Cancer Center, looking at MPG staining to see if there was a correlation. And there was some correlation with both of these patients who had a good progression-free survival with MPG expression. And combination was safe, limited toxicities, so we knew we could, give this combo. What was very interesting, though, was that although the study failed to meet its primary endpoint in terms of there was no increase in response rate, there was no difference in overall survival or progression-free survival. In a select MGMT group of patients, the median overall survival was 30 months. This we know is definitely better than what we have seen. And if you look at it intuitively, it makes sense, right? If you have two hits, if you have an MGMT hit in methylated patient population, so most of the repair will happen through the base excision repair pathway. If you now add a base excision repair inhibitor, this dual approach can be beneficial. And that's what we had saw in this trial. What was also, so the concept was that at least in MGMT methylated patients, this approach is promising. What was very exciting was this work that we had done in collaboration with Dr. Mike Behrens, because at Cleveland Clinic, I had an IRB where we could do profiling of our patients. And two of the patients who did well on this trial actually were the ones I had treated, and we had several samples that we were able to send to TGen. And basically, Dr. Behrens looked at this gene set enrichment analysis, also known as gene set variation analysis. And essentially, this was MCDB, which designated 44 genes for the founder gene sets for DNA damage response. And essentially, these are the two patients who were good responders. And as you can see, they had a particular gene signature. And these were what we termed as exceptional responders because their progression-free survival was really good. In first patient, we had an overall survival of 32 months. Second patient, the overall survival was close to 5 years. And then we had a third patient who, again, we treated at Cleveland Clinic. just progressed before 6 months. So he did not meet that PFS 6 criteria, but he had gotten 6 cycles. So essentially, and this was the borderline signature. So fairly robust data about this premise of this technology. Then we actually looked at in this responder patients to see if there was any response to chromosomal instability signature and proliferation signature. And we did find that in the responders. We then looked at was the response to TRC102 independent of molecular subtypes, and we found that it was. So then what we did was we actually went to the TCGA and looked at what proportion of patients have this DNA damage response signature that we had shown. And here you can look at the far right side, that around 10.5% of TCGA patients have this. And intuitively, it made sense because we had 19 patients and 2 of them had signatures. So that was roughly 10%. So we do know that this genotype signature is present around 10% of patient population. So with that, we actually had proposed a study, which is initially going to be a phase 1 study through the Adult Brain Tumor Consortium. And unfortunately, as you know, that the funding has been limited to that consortium. So we brought this trial to SWOG. And here, the premise is to run the trial, and we're going to do a phase 1/2 study of TRC102 in patients with newly diagnosed MGMT promoter methylation. So it will be a sister trial to the one Dr. Tony Wang had proposed. And here, everyone will get 6 weeks of radiation along with temozolomide. But we will then use TRC102. And essentially, so the patients who will not qualify for that methylated study will be eligible for this. And we do plan to validate the DNA damage response pathway in a prospective manner. And essentially, this will be 3+3 design, but our plan is to do a phase 2 study based on the feedback we got from the Rare Tumor Committee, where they would like us to do phase 2 trials rather than phase 1 studies. And so we have a call coming up with the statisticians, and then our plan is to do again a CTEP pre-submission call so that we can discuss this concept. And a sense is to present these 2 concepts together so that patients, if they have an MGMT methylation promoter, they'll go to this TRC102 study. If they're unmethylated, they'll go to the Toci study that Dr. Wang had outlined. With that, I'll stop, and I'm happy to take any questions regarding this concept. So not hearing any questions, I would then want to move on. Obviously, as a part of this working group, one of the things that I wanted to do was have a robust relationship with basic scientists across the country. And if you have, or if you work with basic scientists who would like to be part of our working group, please send me their email in the chat box. All of those who want to be part of our working group going forward and participate in those periodic meetings, please send me your email in the chat box and I'll add you to the working group list that we are working on. But next, I'm very privileged to invite Dr. Justin Lathia, who's one of my partner in crime at Cleveland Clinic. We worked for over a decade together. He's an exceptional basic scientist and has had multiple R01s and also serves as a PI of a PO1 there. And his lab has been studying cancer stem cells, have been looking at gender-specific and numerous other pathways because our intent is to do more bench-to-bedside trials. That's the way to move scientific progress. And our TRC102 study is a great example of that. We had a biomarker to begin with, and then we learned another biomarker. Otherwise, this trial would have been negative. So with that, I would like to invite Dr. Justin Lathia, and he's going to talk to us about leveraging laboratory observations for the next generation clinical trials.
Okay, thanks so much, Dr. Ahluwalia. Can you guys see my screen?
Yes, we can.
Okay, thanks so much. You know, it's actually really good to be here, and I'm really excited to sort of follow not only Dr. Horbinski, who's really kind of laid down the groundwork of the complexity of the new brain tumor grading system or WHO classification, but you know, it's also great to see in even the presentations from Dr. Wang and Dr. Ahluwalia that you know there's a basic science aspect to it, and I think you know collectively this is a real opportunity to, I would argue, run better trials. So, you know, as Dr. Ahluwalia alluded to, my lab's been very interested in this idea of cancer stem cells. You know, full disclosure, my background is actually developmental biology. I spent quite a lot of time thinking about how stem cells actually function. And then I got very interested in applying this to diseases like glioblastoma. So, this is sort of the way we think about glioblastoma. We think the cancer stem cell is having a variety of bidirectional interactions that are depicted here. And, the labs worked on mechanisms like cell adhesion, cell-cell communication, niche communication, lipid metabolism, and then this idea of the interface between cancer stem cells and the immune system. And I want to really show you, and I should say, anything in purple is on the preprint server. Our lab is very open about science, so please feel free to go to the bioRxiv preprint server. We actually have 3 or 4 unpublished papers that are working their way through review right now that are on there, so we would love to get feedback. But this has really been a launching point for us to think about how do we take these observations in the lab and get them into the clinic. And through our partnership with folks like Dr. Odi, it's really happened. So I want to give you examples about how we can think about alterations in lipid metabolism, as well as myeloid-derived suppressor cells, and how this could potentially be informative for next generation clinical trial design. So, years ago, we really thought about this idea of, can cancer stem cells directly interact with the immune system? And we got really hooked on this subset of cells, or this lineage of cells called myeloid-derived suppressor cells. I'll explain them a little bit more later, but they actually come in 2 subtypes, or at least 2 subtypes, the monocytic and the granulocytic. The monocytic shown at the top, and that can further differentiate into macrophages and dendritic cells, and the granulocytic is allegedly terminally differentiated. So again, as a developmental biologist by training, I'm absolutely enthralled with this cell lineage because I think it really provides an opportunity to understand the intricacies of immune response, especially in glioblastoma, where we know that a lot of immune therapies are not optimal. So again, the lab's been asking questions about myeloid-derived suppressor cells as biomarkers of disease. I'll give you 2 examples in a second. What are the differences in subtypes? I'll show you that a little bit later. How can we target them? I'm going to give you several examples here. And again, this was really multidisciplinary work. Again, a really good team of physicians, scientists, medical oncology, neuro-oncology with Dr. Odi, Dr. Mike Vogelbaum, who's now at Moffitt for neurosurgery. And we've just had a great group of graduate students, postdocs, neurosurgery residents, medical students in the lab over the last decade who have really pushed this forward. So, the other thing I just want to point out is that there's actually a very complex interaction between the stem cell state and the immune system in tumors in general. And this is work that Jeff Nabayek just recently published in Nature Review Cancer. And there's a series of bidirectional interactions. So earlier on, Dr. Wong talked about IL-6, and you can see that's very important for the interaction, for example, between cancer stem cells and macrophages. And my lab's been very interested in this idea of myeloid-derived suppressor cell cancer stem cell interaction through the MIF family. I'm not going to have time to talk about it now, but just be aware there are opportunities to target MIF through a variety of ways, including ibutylast, which is a compound made by a company called Medicinova. And that's really just finished phase 1 clinical assessment at Dana-Farber, and Patrick Wen is the PI on that. So in terms of these cells, these myeloid-derived suppressor cells are immature cells that originate in the marrow and get aberrantly activated during a variety of conditions. So for example, tumors, pregnancy, wound healing, obesity, and even COVID. And what happens is these cells traffic to sites and are potently immune suppressive, mainly to T cells as well as NK cells. And as a field, there are a variety of outstanding questions. There are some that would still argue, look, these are just aberrantly activated monocytes and neutrophils. We do think that they're actually quite distinct. And we're really trying to understand the last 2 questions on the bottom left. What's their function in cancer? And how can we actually target them? I will say we've done some single cell sequencing to really dive in to this problem. And it's no surprise to us. We obviously see a vast amount of heterogeneity within the populations. And just to orient you, population 1, hopefully you can see that, that's actually classic macrophages. So we do think that they're different than macrophages. And again, this was initial work done with Antonio Ivarone and Peterson's at Columbia. So we think that this population is still to be determined, and it is quite complex. But what do they look like in patients? So what we and others have shown is really this graphical abstract that was published from a PhD student of ours, Tyler Albin. And really what we tried to make an argument of is in glioblastoma patients, there is just a higher level of these cells, not only in the tumor microenvironment, but also in the circulation. And if you look at patients that have a good prognosis, you actually see that there's less of these cells in the tumor microenvironment and in blood over a function of time. And you see the diametric opposite when patients have a poor prognosis. And then in collaboration with Alvaro Alvarado, who was a PhD student with our group and is now at UCLA with Harley Kornblum, we use next generation technology, mass cytometry time of flight, otherwise known as CyTOF, to reconstruct the immune system. And what we found is actually that patients that have a good prognosis actually have a signature where it looks like the myeloid-derived suppressor cells have differentiated into dendritic cells. So we've been trying to target this in a variety of ways, and this is by no means specific, but what we have found is low-dose chemotherapies, such as 5-FU, can actually do this. So here's a preclinical experiment Balint Otvos in the lab did over 5 years ago, where we started transplanting in a syngeneic mouse model system and giving low-dose 5-FU once a week. And what we were able to find is that in the circulation, we were able to reduce the myeloid-derived suppressor cell population. If we luciferase tag our tumor cells, so these are the GL261 model that Dr. Wang mentioned earlier. Again, you see a reduction in the signal. And again, it's really a dosing sweet spot. So it turns out that if we go at about 10 mg per kg in this mouse model, we can actually get a group of mice that are refractory to the tumor. And if you actually look in the microenvironment, as you would predict, as you reduce MDSCs, you should actually see an increase in intratumoral CD8 positive T cells and a reduction in Tregs, and we see that. Now, I want to be clear that this is not an MDSC specific therapy, because if we go to a higher dose, we actually kill the T cells as well. So it's sort of this sweet spot that we're trying to find. And this was the basis of a recently completed clinical trial that was run by David Peerboom at our center. So again, this was published in 2019. You're welcome to look at the paper. But what we actually noticed is that when we looked at MDSCs as a function of time, and this is in the black group is a control group, and each sort of different shape is a different blood draw. So if you look at the peripheral blood of these patients, over time in general, the MDSC levels go up. But as we increase our dosing, and here we use capecitabine, so this is the prodrug for 5-FU, you see a reduction, and that's quantified here. So this gave us at least a first glimpse that in human patients, we could actually develop an approach to reduce MDSCs. We've done CyTOF as well on these patients, on their tumors, and we see a similar signature. But I want to again highlight the fact that MDSC monotherapies may be useful, but they should also be thought about in combination. So here's an example. If you look in the left in advanced melanoma, it turns out that patients that are refractory to conventional checkpoint inhibitors actually have an increase in circulating MDSC levels. In collaboration with Dr. Alawalia in an ongoing clinical trial, we see the same thing. So again, it's just an idea that we may be thinking about developing anti-MDSC therapies, but we've got to think about it in combination with other approaches. So again, I just told you about using low-dose 5-FU. Can we do this in a little bit more of an educated way? And we in the lab have been thinking a lot about these myeloid-derived suppressor cells and their subsets. This is a paper that was published last year. And as Dr. Alawalia also mentioned, it's actually part of a program project we have that he's a part of from the clinical perspective, where we're really interested in sex differences. So I'm showing you the punchline first. When we tried to answer the question, what was the difference between the subsets, Defna Bayek in the group, who's shown in the bottom right, actually realized that the monocytic subsets were flooding the microenvironment of males. The granulocytic subsets were in the periphery of females. So we started observing this potent sex difference, and we really kind of ran with it. So what we did was, here's just initial data showing that in our preclinical models, whether we use GL261 or SP28, this is another black 6 model that's generated through the Sleeping Beauty system given to us by Hideo Okado at UCSF. We actually see that same cell, again, the only difference here is male or female host. You actually see a difference. The females tend to do better. We think this is immune driven because we can reverse this through bone marrow transplantation. And then what we kind of realized is that these sex differences weren't only in mice, we see them in humans. So those of you who treat patients may be familiar with these incidence curves. There's two groups. There's a blue group and a red group. And really the difference is actually not mutation. It's not environmental factor. It's biological sex. And Jill Barnholt Sloan published a paper a couple of years ago where, using the Ohio brain tumor study, she made an argument that if you adjust to all clinically relevant variables, males have a poorer prognosis. And other members of our program project group, led by Josh Rubin, really looked into this at the transcriptional level. So I invite you to look at this paper. There's a lot of data here. But one of the things that Josh's group realized when they did a large-scale drug screen is that there's actually differential response, whether it's in mouse models or in human patients or human cell lines, to different drugs. And for example, it turns out that female tumor cells are actually more responsive to temozolomide. So maybe that's something that's underlying this sex difference. I don't know. But we've been interested in this in the context of immune response. So going back to our mouse models, what Daphne did was she transplanted, and we started depleting myeloid-derived suppressor cell populations peripherally. And really, we were only able to see the extension of survival in female mice. So whether we go after the entire subset using GR1 or the granulocytic subset using Ly6G, we can extend survival. So what we started doing was RNA sequencing these cells directly out of our mouse models. And the honest truth is we did this to generate reporter mice for future mechanistic studies. We've actually struck out. We haven't been able to do that. But what we actually did was we were able to use a drug prediction algorithm generated by Fei-Shuang Cheng, who's also in our institute. He's actually 3 floors up from me. And what Fei-Shuang was able to do was take our RNA sequencing signatures and map them on drug response profiles. And essentially what he was able to do is predict, for example, that males should be sensitive to low-dose chemotherapies. So fluorouracil is what we used almost 5 years ago. Capecitabine was what was on our clinical trial. But interestingly, females, and especially the granulocytic subset, were acutely sensitive to IL-1 beta inhibition. Right, so again, these are drug prediction algorithms. The proof is in the pudding. So it turns out we did the preclinical experiments. So we started transplanting, again, same cell, only difference is male or female host. Whether we use fludarabine to go after monocytic MDSCs, we only see a survival signal in males. And what's interesting is if you antagonize IL-1 beta, which should work on granulocytic MDSCs, you only see the signal in females. So at least this is our first indication that from an immune therapy perspective, there may be an opportunity to leverage sex differences. So that was all published. So what we've been working on now is we're trying to still understand the difference in the subsets, because again, I gave you examples of IL-1 beta and low-dose chemotherapy, but I think actually there's another opportunity for additional molecular targets. So what we then did was we went back to our systems and we started adoptively transferring myeloid-derived suppressor cells. So what we basically do is we generate syngeneic mouse models, and then we adoptively transfer different subsets. And all the data I'm going to show you is in male mice, but essentially if we start adoptively transferring, it's really the monocytic MDSC subset that seems to be driving tumorigenesis, not as much the granulocytic subset. So that really tipped us off to the fact that there's likely something cell intrinsic. How do we deal with that? So we teamed up with Pete Scaccieri, who's one of our colleagues at Case Western through the Case Comprehensive Cancer Center. And we actually did ATAC sequencing to try to understand, are there regions of open chromatin that may be informing the difference? And our smoking gun was really the fact that there was huge differences in the monocytic subset with adhesion genes. So we went ahead and validated this in mouse models. So here's just examples between the subsets. Blue is the monocytic, pink is the granulocytic. And hopefully you can appreciate that, at least in the marrow and the blood, there's more integrin beta 1. You see the same thing with beta 7. And what's interesting is if you look at the function of beta 1, and this can be read out through the activation state using a conformational specific antibody called HUTS4, you see the same thing. So monocytic MDSCs had just a more activated beta 1 integrin level. We validated the same thing in human samples. So this was in collaboration with Matt Grabowski and Ali Rezan Mohammadi on our neurosurgery team, who were able to provide matched blood and tumor from the same patient. And you can see the same thing. And actually Filippo Veglia and Mike Vogelbaum at Moffitt have also seen literally the same results. So it seems like in human patients, the monocytic MDSCs have a higher level of integrin beta 1. And again, with Yifan at University of Pennsylvania, we validated this bioinformatically. Okay, so I'm telling you that fine monocytic MDSCs have a higher amount of integrin beta 1. And this actually may be why they bind to the tumor microenvironment, but this isn't functional at all. So we went back to our mouse models, and prior to adoptive transfer, we neutralized the function of the integrins with blocking antibodies. And really, I just want you to take a look at the top Kaplan-Meier survival curves. Essentially, we can dramatically blunt tumor growth if we neutralize beta 1 prior to adoptive transfer. The time frame we used to neutralize beta 1 was not enough to induce cell death, so it wasn't like we were putting in dead cells. We were actually preventing additional myeloid-derived suppressor cells from getting into the tumor microenvironment. And here's just an example of some profiling that we've done that really kind of confirms that. And what we actually think is happening is when a lot of our monocytic MDSCs are getting into the microenvironment, they're transitioning to a tumor-associated macrophage stage, which can still be immune suppressive. And that's really blunted when we neutralize integrin beta 1. So really, here's the graphical abstract of a paper that is currently on the preprint server and working its way through the gauntlet of publication. But really, we think that the subsets are now providing another opportunity for custom therapies. And again, we're working on signaling validation, and we're really trying to work our way through using preclinical proof of concept compounds that may actually be the basis for additional clinical trials. So again, this is what we're doing right now. I should mention that Dr. Ahluwalia, in collaboration with Novartis, is going to be starting a clinical trial antagonizing IL-1 beta. I didn't have time to talk about it. We also are doing a lot of combination therapy studies with CAR T cells with collaborators in Israel. So that's Denora, as well as Joost Mellenhorst, who's at University of Pennsylvania. Obviously, we're very interested in just the clinical therapy response. We also are very interested, as I told you, in the lineage relationship, and we are certainly expanding these sex differences to other immune cell populations. It's a little early now, but in our preclinical models, we are seeing profound differences between males and females in different lineages of T cells as well as macrophages. So hopefully within the next 6 months, we'll be able to finalize that and sort of share those data. I just want to end with a very provocative idea of trying to think about other factors that could be driving glioblastoma growth and how do we target them. And really, this is a meta-analysis. Just like nearly every other cancer, obesity seems to be a risk. And this is just a meta-analysis trying to make an argument, at least in females, that obesity may be a risk for glioma. Now, a lot of this is complicated by the fact that the body mass index is not sort of gender neutral. There's a bit of a bias there. But all of that aside, we tried to model this. So this is work that was done by Dan Silver that was published several months ago in the Journal of Clinical Investigation. And he really came at this in a different way. So he started putting mice on a high-fat diet. And I will tell you that there wasn't a huge difference in weight when we do this, but we get sort of high-fat diet, and we can basically speed up tumorigenesis in these mice. It's not an obesity thing per se, because we do the same thing using OB mice, which have alterations in leptin signaling, and we actually don't see a signal. But what Dan was able to work out is the fact that when you put mice on a high-fat diet, and that's shown in the right panel, what ends up happening is we end up plummeting hydrogen sulfide in the tumor microenvironment. So hydrogen sulfide is a gasotransmitter, which can actually sulfhydrate proteins. So this is just like phosphorylation or nitrosylation. Proteins can also be sulfhydrated. So essentially what happens is we plummet hydrogen sulfide levels, and that actually drives tumor growth. And in the paper, we were able to rescue that preclinically. I should mention that hydrogen sulfide reduction is also a hallmark of aging. So in collaboration with Derek Wainwright at Northwestern University, we're trying to understand the nexus between this and immune response. But essentially, hydrogen sulfide, we think, is functioning as a tumor suppressor in glioblastoma. So it begs the question, can we actually reverse this? There's a lot of ways to do this, and none of this is specific. I just want to be upfront there. But for example, there are ways to do this. There's a compound called PTU, and this was actually an example of a clinical trial run in 2003, actually out of the Cleveland Clinic, by Jean Barnett, who's the director of our center, and was actually able to show that we can boost H2S. We're revisiting this hypothesis. They were looking at it in the context of hyperthyroidism, but we're now starting to screen a variety of agents that are clinical grade for the ability to boost H2S, just as a potential, again, it's a little bit of an off-the-wall idea, but just another way to sort of go after GBM. So I want to stop there, thank the folks in the lab. So this has been us over the last 10 years or so. I've listed all the people on the right. And I want to thank the great collaborators we have internally throughout the US and the world, and the funding agencies, and anything sort of in purple or orange is really directly related to what I just told you. So I want to thank Dr. Ahluwalia again for sharing this with you. And hopefully this can get the conversation started with some additional concepts.
Thank you so much, Justin, for a very comprehensive overview of what your lab has been focusing on for the last decade. We sincerely believe that the further inroads or improvements in outcomes for glioblastoma is only going to be possible through bench to bedside studies and then back to bench perspectives. We've run countless trials which have failed to reach their primary endpoint. So we have to learn from every study we do and every patient that we treat. So thank you so much. With that, I would open the floor for any questions for Dr. Lathia, if anyone would like to. If not, thank you so much, Justin. Again, good luck with your experiments. We certainly hope to leverage the findings from your lab directly through the SWOG mechanism. I think we have a great group here, and hopefully we can continue to push these concepts through CTIP.
No, I really appreciate it. And I put my email in the chat as well. Please feel free to follow up. If I can convince any of you to think about a crazy hydrogen sulfide boosting strategy, I would love that.
No, absolutely. And I am noting also people's emails when they're putting it in the chat box. And so we're going to shift and change gears. We heard a lot about studies and concepts and research focused on gliomas. We're going to move to CNS lymphoma. With that, I would like to invite Dr. Yasmin Odia, who's the chief of Neuro-Oncology at Miami Cancer Institute and a colleague, to talk about her concept on phase 1/phase 2 of pemetrexed plus Selenexor for recurrent refractory CNS lymphoma.
Thank you for the opportunity, and thank you, Dr. Ahluwalia and the SWOG team for considering this protocol. This is a truly rare entity, and I think an entity that also needs further investigation to improve outcomes. I will discuss here a phase 1 and phase 2 trial of Selenexor plus pemetrexed for relapse or refractory primary CNS lymphoma. These are my disclosures, none of which are relevant for today's discussion. I like to recognize the team members of this protocol that include Dr. Ahluwalia, as well as radiation oncologist Dr. Hall and Dr. Linares and the BMT group. In terms of background, we know that despite initial high-dose methotrexate, which remains the standard of care for frontline primary CNS lymphoma, many patients, in fact, relapse and even die from primary CNS lymphoma. There is no standard of care in progression or relapse. There are numerous small pilot studies and early phase studies that serve as an impetus for treatment, but no real standard of care exists. That being said, pemetrexed is an NCCN approved salvage therapy for these patients. And I'd like to introduce an agent, Selenexor, which is a first-in-class small molecule inhibitor of exportin. It has known activity in refractory relapsed systemic diffuse large B-cell, in fact, receiving FDA indication for monotherapy, and represents the overwhelming histological subtypes of primary CNS lymphoma, in fact, about 90% or more. Selenexor also has established CNS penetration, all of which I will provide data for. And therefore, we argue the Selenexor efficacy in primary CNS lymphoma warrants further study. In terms of pemetrexed, which is the background and the comparative group for this trial, is a multi-targeted antifolate that has some similarities to methotrexate, but with some broader spectrum of activity. It already has shown single agent activity in relapsed refractory primary CNS lymphoma. We will use this as a historical cohort, and we'll present the data for the study performed by Razor et al. In this cohort, the median age was 69.8, so typical for our patients, which are a bit advanced. Median KPS is 70%, but they did have a looser KPS criteria. And 10 out of the 11 had already failed high-dose methotrexate. Methotrexate in this regimen is actually dosed higher than the typical lung dosing. It's 900 milligrams per meter squared every 3 weeks, along with low-dose dexamethasone, folate, and B12 supplementation. Of note, lung dosing is usually around 600 milligrams per meter squared. The treatment is given in 6-week cycles and continued until complete remission of progression or toxicity as defined by the study. And a median duration was 5 cycles or 10 doses. The overall response rate in this group, in this trial, was 55%, with a disease control rate of 91%. The progression-free survival at 6 months was 45%, with a median PFS of 5.7 and a median OS of 10 months. The toxicities were primarily hematologic and infectious, similar but to a higher frequency and severity than non-small cell because of the higher dosing. Progression-free survival and OS curves are provided here, and the toxicity profile on the right, in particular the grade 4 toxicities mainly being pancytopenia and 1 death due to infection. In terms of Selenexor, as we discussed, it is a first-in-class oral SINE inhibitor. It is orally administered anytime between 1 or twice weekly, depending on the regimen and in the trial. It has no known drug interactions. This we validated with a pharmacy consultation. There's already been over 3,200. I'm sure this is out of date, so there's probably many more of patients treated with Selenexor alone or in combination with various chemotherapies across multiple tumor types. The anti-tumor activity is being studied even more in advanced hematologic and solid tumors. And the main side effects, which are anorexia, nausea, fatigue, and platelet reductions, are often manageable with standard supportive care, including steroids. Treatment has even lasted over 2 years in patients without a severe cumulative or irreversible organ toxicity. It is already approved as the first ever approved indication for this drug was relapsed refractory myeloma after 4 lines of treatment. And that last statement is out of date because, in fact, it did receive indication for recurrence diffuse large B-cell lymphoma. Again, the mechanism of action is really inhibiting transport between the nucleus and cytoplasm. It seems to have a predilection regarding tumor suppressor proteins, and in particular seems to have a therapeutic window focused on G1 and G2 arrest. It seems to have a wide spectrum of tumor efficacies, and as it really targets cell cycle progression, apoptosis, and metastasis. It has a clear linear PK, and furthermore has proven CNS efficacy in preclinical models, and we will show also that it has CNS penetrance in human clinical trials as well. This is kind of the landscape of the drug development, and again, there's now a 2021 that has additional indications. As I've said, it's been studied across multiple hematologic and solid tumors. I will highlight only those that are relevant for the topic today. In particular, there was the SADAL study, which was Selenexor monotherapy for relapse diffuse large B-cell lymphoma given at 60 milligrams twice daily until progression or tolerability. Again, the grade 3 to 4 toxicities were largely pancytopenia. However, they also did witness sodium decreases as well as other electrolyte abnormalities in terms of laboratory abnormalities. Fatigue and anorexia were also toxicities, but not listed on the table here of laboratory abnormalities. In terms of outcomes, here we have a survival probability and response curves for these patients. Again, as a monotherapy showing efficacy in this disease histologic subtype. They concurrently had done a study in relapsed glioblastoma, where they again looked at monotherapy, and it was a dose escalation, dose finding trial. that ultimately settled on a dose of 80 milligrams once weekly, again, oral to be dosed in these patients because it had similar toxicity profile to other treatment schemes and had the best in terms of efficacy. I present here some of the response curves as well as the progression-free survival. That is what prompted the current and open and accruing trial in as Export GBM 029 that looks at combinatorial treatment. I highlight for you arm B, which is newly diagnosed GBM for MGMT methylated subtypes, that in fact is Selenexor plus temozolomide plus radiation, followed by adjuvant Selenexor plus temozolomide. They have multiple arms that include combinations with bevacizumab, lomustine, and radiation alone. But I highlight temozolomide as often that is a therapeutic agent we do use in lymphoma. In terms of the clinical experience for primary CNS lymphoma, unfortunately, there has not been a clinical trial to date, and hence the impetus for this trial. However, there has been off-label use that has been reported as a case report in a single patient that in fact showed activity on diffuse large B-cell systemically, and therefore a 55-year-old man who had had multi-recurrent systemic diffuse large B-cell lymphoma, germinal center type, had received and failed after R-CHOP and various other treatments, including transplant with beam preconditioning. He in fact had a CSF and ultimately a CNS recurrence, and it was for this indication that he received expanded access to Selenexor, dosed in the diffuse large B-cell scheme of 60 milligrams twice weekly. So this patient had a marked response to single agent Selenexor, again in an expanded access form, had two lesions in particular, one that had a complete remission and the other one that had a near complete but therefore partial response. Ultimately, however, grade 3 toxicity and anorexia led to treatment reduction and hold, and ultimately recurrence and terminal death from a recurrent CNS lymphoma. So we introduced our concept, which is a phase 1, phase 2 trial of Selenexor plus pemetrexed in the relapsed refractory primary CNS lymphoma. Pemetrexed, again, dosed at least the target dose is 900 milligrams per meter squared every 3 weeks, with a concurrent dosing of Selenexor using the GBM or CNS penetrating regimen of 60 to 80 milligrams once weekly, to be followed by ideally a consolidation recommended but not mandated of either a transplant based on clinical criteria. There's different intensity of preconditioning, or whole brain radiation if needed for residual disease. A maintenance Selenexor regimen is really only advised in the whole brain or non-consolidation therapy groups. And really our focus in terms of response and endpoint will focus only on the induction phase. And so I detail some things below. The typical workup and baseline assessments for primary CNS lymphoma are slit lamp exam, PET CT, CSF, and repeated if there's any evidence of positivity of any of these at baseline once patients have completed induction. Again, this is a phase 1 trial, so we'll have a 3+3 design to start, and we'll explore 2 Selenexor dose levels starting with 60 and then increasing to a target goal of 80 milligrams once weekly if tolerated for a maximum of 12 patients in the phase 1 component. Pemetrexed for the first dose for cycle would be given 800 milligrams per meter squared. If tolerated, then subsequent doses will be given at 900 milligrams per meter squared until progression, toxicity, or death. Brain MRIs are every 6 weeks, with including after transplant, and then every 4 to 6 months as clinically indicated in the post-transplant or post-consolidation setting. Again, the consolidation regimen will provide guidance and recommendations, but it's ultimately left to the discretion of the investigator. And for patients not receiving transplant, those patients are eligible for a maintenance Selenexor of 6 months. The statistics, again, focuses on the induction phase. So we have a single arm open label phase 2 trial with a lead in phase 1 trial. The analysis include all patients treated at the recommended phase 2 dose, whether that's the 60 or the 80 milligrams of Selenexor. And we design a one-arm binomial using the null based on the RAZOR study of an ORR of 55% with a target goal ORR of 75%, a power of 80 and alpha of 5. This meant that we needed 35 eligible patients and 40 to be enrolled, considering a 15% attrition. And again, the ORR is only based after the induction and not subsequent phases of treatment. We would be using the standard response criteria of the International Primary CNS Lymphoma Collaborative Group. And we would be actively and proactively managing the expected toxicities from Selenexor that include anorexia, nausea, hyponatremia, fatigue, and cytopenia, with clear guidance as to supportive care. With that, I welcome your questions.
Thank you so much, Dr. Odi, for a very comprehensive overview of your concept. With that, I would open the floor for any questions. While people have questions, I wanted to ask you the degree of interest on the part of the company to support this concept through CTEM.
Very interested. They are more than willing to furbish a letter of support. They had already approved the concept in terms of a concept of an LOI. So in reality, the floor is on our court.
Okay, so I think the next steps, based on my prior interactions with SWOG, is going to be to present this concept with the lymphoma group, because since this does transcend, two working groups or committees, so I will be in touch with you and try to see if we can get on the agenda for the next lymphoma committee and meeting, and put the present the concept there. And if that would be of interest, I think, well, we can certainly have a CTEP pre-submission, again, a call, since the relatively size of all these 3 trials can actually go through just CTEP and avoid the brain malignancy steering committee review. So I think certainly we don't have a SWOG CNS lymphoma concept right now. So I think this would be of interest to us in the group. Would there be any questions for Dr. Odi at this time point? If not, I know we have around 18 minutes still left in our time. I know Dr. Chad Rastovan has been an integral part of our working group and has been doing some really amazing work in radiation oncology. He's got interest in both CNS as well as lung. And I did present the Maverick trial that he's spearheading nationally on behalf of SWOG in collaboration with Dr. Paul Brown. He did have a couple of studies in brain mets that he's been championing and he's been part of. So Chad, if you want to use the next, probably, if you would be okay, probably next 7-10 minutes. And then that will still leave a little bit of time for open discussion, because I really want to leverage the group and see what else can we do to, move and gather more momentum for our working group.
Great. Thank you so much for the opportunity. My name is Chad Rastovan. I'm a radiation oncologist at the University of Colorado. And I'll just be going over a few studies that are championed in SWOG. So this study is NRG CC009. And this is a phase 3 trial in patients with small cell lung cancer brain metastases, randomizing patients to stereotactic radiosurgery versus hippocampal avoidant whole brain radiation. The PIs are Dr. Vinay Gandhi, and I serve as the co-PI on the trial with this great group here. So for background, as many know, whole brain radiation is still the standard of care for small cell brain metastases. Even for patients with a single brain metastases, it's technically the standard of care. Prior trials that have helped to establish radiosurgery alone have excluded small cell patients systematically. And historical objections to the use of radiosurgery in small cell have primarily been related to a concern for short interval CNS progression in some patients, which could potentially reduce overall survival outcomes. However, we also have learned over the years that whole brain radiation can be associated with toxicity to both cognition and quality of life. And there are various strategies to mitigate this, including the use of stereotactic radiosurgery alone. So concurrently, there's been some additional evidence that's been put out on the topic of radiosurgery alone in small cell brain metastases. This study that we're showing here is the FIRE SCLC study, and to date, this is the largest study on the topic. It's a retrospective, multi-center international study. Ultimately, there were 710 patients who were treated with first-line radiosurgery without prior PCI or whole brain radiation. And these were compared in a propensity score matched analysis to patients who received the standard of care, which is upfront whole brain radiation. And what we found was really similar results in small cell to what we see in other histologies. So if you look at kind of the primary graphs on the bottom right is the CNS progression graph, and patients who were treated with whole brain radiation had more durable CNS control, longer time until new lesions emerged. But on the left, we have the overall survival curve, and basically what we see is that the better CNS control with whole brain radiation didn't appear to translate into an overall survival benefit. In fact, in this retrospective, radiosurgery looked a little bit better, but we were cautious not to overinterpret that because of the potential for residual confounding in a retrospective analysis. Currently, there's also been data that's helped move the ball forward in terms of reducing toxicity with whole brain radiation. So sequential NRG, RTOG studies have shown that the addition of memantine can improve cognitive preservation, and then the addition of hippocampal avoidant whole brain radiation with memantine can improve cognitive preservation even further. So that leads us to the schema slide for this trial. So this is the phase 3 trial of SRS versus hippocampal avoidant whole brain radiation for 10 or fewer brain metastases from small cell lung cancer. It was activated earlier this year. We have basic eligibility criteria, but the core of the study is quite simple. It's a straight randomization to stereotactic radiosurgery versus hippocampal avoidant whole brain radiation. Accrual goal is 200 patients with a primary endpoint of time to cognitive failure using standard cognitive testing battery. And of course, we'll have other important outcomes like overall survival and time to CNS failure as well. So to our knowledge, this is the first phase 3 randomized trial looking at SRS alone in small cell lung cancer, so it's very exciting. Activated earlier this year, it's accrued 9 patients for a goal of 200. And so if this trial is attractive to you and you think that you could accrue patients at your center, please feel free to reach out to me. We'd be happy to work with you to try to get it open. We do think it's a really great trial to be able to offer patients. And I would just mention too that it's integrated well with the Maverick trial that Dr. Odi presented earlier. So you'll recall the Maverick trial is a randomization of MRI surveillance plus or minus PCI, and patients who develop brain metastases in the MRI surveillance arm can go on to the NRG CC009 trial for their treatment. So here's just the schema for the Maverick trial that Dr. Odi presented earlier. And so again, this is limited and extensive stage small cell without brain metastases, randomized to MRI surveillance plus or minus PCI. And this trial is also exciting. It's the first randomized trial looking at MRI surveillance plus or minus PCI in limited stage, and it kind of represents a tiebreaker, if you will, for the conflicting overall survival outcomes with PCI in extensive stage. And it's designed to have very low barriers to accrual. It allows hippocampal avoidant PCI. And so if this is interesting to you too, please feel free to reach out to me. I'd be happy to work with you to try to get it open and help us answer this very important question that affects quality of life and cognitive function in small cell patients. Moving on to 2 other endorsed trials. So the Alliance A07-1801 trial is led by Paul Brown. This is for patients with resected brain metastases who are going to receive post-op radiosurgery to the cavity, and it's a randomization to single versus multi-fraction radiosurgery. So the background is that post-op radiosurgery to the cavity is now considered a standard of care. It does improve local control outcomes, which has been demonstrated in randomized trials. However, the local control outcomes with single fraction radiosurgery have been a little bit suboptimal in prospective studies, and there's non-randomized data suggest that multi-fraction radiosurgery may be better in terms of local control and possibly toxicity when we treat the cavity in a multi-fraction approach. So this study is testing that. So this is for patients with brain metastases that are resected, and then it's a straightforward randomization to stereotactic radiosurgery to the cavity using either a single fraction or multi-fraction approach. The primary endpoint is time to surgical bed recurrence. The inclusion criteria: the metastases need to be greater than 2 centimeters. Patients cannot have had prior whole brain radiation for this study. This study was activated at the end of 2019. Target accrual 208. It's accrued extremely well. The accrual as of this morning is 147 patients of the 208. It was endorsed by the SWOG CNS Working Group earlier this year, and we're very excited to help contribute to this trial that looks like it's going to be very successful in answering this question. In talking with Paul Brown, the PI, he just asked me to thank everyone for participating and to remind sites that please upload the MRI imaging because the primary endpoint is a centrally reviewed image-based assessment of surgical bed control. And that leads us to the final study that I'll present, NRG-BN009. This is a phase 3 trial of salvage radiosurgery or salvage radiosurgery plus hippocampal avoidant brain radiation for first or second distant relapse after upfront radiosurgery in patients with brain metastasis velocity greater than 4 per year. It's led by Dr. Michael Chan from Wake Forest and Dr. Vinay Gandhi. So the concept is interesting. So there's this concept of brain metastasis velocity, which is an important prognostic factor in patients with brain metastases. And basically, you calculate that by the number of new brain metastases that develop per year. And this is specifically looking at patients who have had upfront radiosurgery. So this is in the relapse setting. And there's been a number of studies that have shown what these graphs show here, which is that the rate of developing new brain metastases appears to be prognostic for both overall survival, but also neurological mortality. So if you're developing brain metastases quickly, you're more likely to die of neurological death. And so that raises the question, should we be altering our therapeutic paradigms, maybe being more aggressive using whole brain radiation in these patients? And so that leads us to the schema. So it's a phase 3 randomized trial looking at patients who've had prior radiosurgery and then have had a recurrence with a brain metastasis velocity of greater than 4 mets per year. And these patients are randomized. All patients will receive stereotactic radiosurgery, and the randomization is plus or minus hippocampal avoidant whole brain radiation. The primary endpoint is time to neurological death, which has a very rigorous definition for the study. This is the key inclusion criteria. It includes patients with lung cancer, melanoma, breast, renal cell, and GI, and no prior whole brain radiation or PCI. It was activated earlier this year and is endorsed by the SWOG CNS Working Group as well. And with that, this is my contact information. Please feel free to reach out to me if I can help answer any questions. And thank you, Dr. Ahluwalia, for the opportunity.
Thanks so much, Chad, for sharing all these trials, and I would like to congratulate you on really doing some amazing work both in SWOG but also working with other cooperative groups. I think that's one of the key components of the cooperative groups is how not only to push trials to our own group but also how to collaborate with other colleagues. I know we still have 7 minutes. We can certainly end early, but I would at this time point I would would like to open the floor for any general comments. Certainly, as I said, our expectation is the next meeting in spring will be in person, but we would like to hold periodic meetings going forward since we at least have 3 concepts which look like is something that we can push through this group. So we would have constant interaction, and obviously would love to get people's input how to make these concepts better, but also to push concepts along, which will help facilitate our working group in its journey to being a committee, which will then leverage even more resources. I did see a number of you had put in your email in the chat box, so I'll make sure those people get emails about our working group. And if you have friends or colleagues of who would be like to be part of that working group, we'll be happy to involve them. not hearing any comments, I will again like to thank all the speakers, but also very importantly, all the participants for coming to our meeting. And I'll give you back your 5 minutes for catching up with other things. Have a wonderful weekend, everyone, and hope to see people in person in roughly 6 months.
Wonderful. Have a good one.
Thank you.